Okay, well, so this is part two of Introduction to Zoology. It's going to be shorter than the first part. This is where we left off. Constructing phylogenies. I said these are some of the major animal features that they look at in order to group organisms into taxa. Okay. I mentioned these. So we'll go over each one of these individually now. Okay. Here we go. Starting with symmetry. What is the body symmetry like? Well, porifera, sponges is an animal, amazingly. They don't have any. There's bilateral symmetry like us, where there's two halves that are same, and there's radial symmetry, circular, and then there's organisms like this that don't have either. So we call it A means without. So asymmetry means without symmetry. Okay, display, asymmetrical body shapes. Not true multicellular animals. They're the simple, we call them as a colony, a simple multicellular or a colony earlier. Are aggregations of different cells, but no true tissues, no original germ layers. We haven't got to that yet. And it's just the one phylum, the sponges, the phylum porifera, like pores. They have little pores in them, right? Sponges have little pores in their wall. Porifera. They're asymmetrical. Then, I lost. What did I lose? Oh, okay. Then there's radial symmetry. Not sure why I lost it there. Radial symmetry is when the organisms are circular. You look at this. With a circular organism, you can divide them into lots of identical halves, like this. You see radial symmetry. Okay. Then that's Cnidaria. So the next phylum up, a little more advanced, Cnidaria, the jellyfishes and sea anemones. It says organisms, the advantage there is organisms can address the environment equally from all directions. Equally from all directions. Okay. And then here's bilateral symmetry on these organisms. That's all the rest. So that would be like this. If you draw there you go, right there. You got this young lady here, right down the middle of her nose there. You've got two equal halves. Okay, let's turn this light down again here. Make it dark enough so you can't see me. That make it easier on you. Anyway, so only two mirrored halves seem from one division in a sagittal plane, down the center. Well, sagittal doesn't technically have to be down the middle, mid-sagittal. This is a mid-sagittal plane. So down the middle here, mid-sagittal, down the middle here, mid-sagittal, this side are the same. Like that, there's a mid-sagittal plane. Okay. All right. So symmetry was one of the first major features they look at for relatedness. A second feature is how the embryos develop. And this is this particular feature is in regards to the tissues that first develop in an embryo. In the less advanced organisms, the embryos have two distinct differing tissues. They're called germinative layers, to germinate from. Germ layers, and when we call it an endoderm and an ectoderm. They've got two, and then all the other tissues are derived from those. All other adult tissues are derived from those two, called a diploblastic organism, right? di is two, bi and di both mean two. All adult cells are organized into tissues and originated from two original embryonic germ layers, only found in Nidaria. So now we had asymmetry in the periphery. Now we move up to Nidaria, and we saw their they have radial symmetry, and they have embryonic tissues that first developed two types only found in the Nidaria, jellyfish and sea anemone squirrels. But then the rest of the organisms that we're going to look at are triploblastic organisms. So that means that their embryo, that little blastosphere, it's like a little glob of, the first glob of cells that develops as an embryo, blastomere, blastosphere, well, either way, blastosphere. Within that embryonic glob of cells, there's three, they can identify three different tissues now, a little more advancement. So that means that more adult tissues can be derived from that, from three rather than being derived from two embryonic distinct germinative tissues. So we've got an endoderm and an ectoderm like we did before, but there's a mesoderm in between. Cells are organized into tissues in adults and originated from three embryonic germ layers now rather than two. A little more advancement includes all the bilaterally symmetrical animals from the. So as we go up from the Nidaria, then the next one we'll look at are the flatworms, Platyhelminthes, on up to the primates, so which are chordates. So all the organisms there from then on will be triploblastic organisms. So the periphery are not considered either. They don't have that these features, these embryonic germinative features. They don't have that. So here, if you look at this, actually, here's our blastosphere. And we've got this, this is the ectodermic tissue. There's the endodermic tissue and mesodermic tissue. Okay. All right. Another major feature zoologists look at is the organism a protostome or a deuterostome? Pro is before, right? So the protostomes came first. Deuterostomes came later. Although that's not what it refers to. Now I think about it. The protostome part is before mouth. Stome is mouth. And deuterostome is second mouth. First mouth, before mouth, first mouth, second mouth. So I'll show you what that means. So protostomes have certain features and deuterostomes have certain features. Let's see what they are. Fate of the blastopore. So what that means is if we look back at this embryo, a pore forms first. It's called the blastopore. It's like an opening. It's going to become the mouth and another opening, eventually become an anus if they have two openings. But there's the mouth opening. So in a protostome, that first blastopore becomes a mouth. Whereas in a deuterostome, it doesn't. The first opening becomes the other end, the anus, and then the second opening, deutero, the second opening becomes the mouth. You see? Cleavage patterns. How do the cells divide in the embryo? Let's see if we even do that. Oh, so here's the fate of the blastopore. And this is showing you that the first opening is going to end up becoming a mouth, and then the anus forms from the second opening on a protostome, and vice versa with the deuterostomes. And then the cleavage patterns are this here. The way the cells in the embryonic glob, the way they divide. Let's see. Radial and spiral cleavage developmental patterns. Radial is when cells line up like this. As they divide, they still are lined up. You see this one over here. And then radial are where the cells, when they divide, they rotate and then rotate. So they end up kind of spiraling. Radial cleavage patterns. So let's see here. Protostomes exhibit spiral cleavage. Um, Radial is, I'm sorry. So did I say, so radial is when they divide straight up, and spiral is, I think I said it backwards the first time. Radial is when the cells divide and they go straight up. Spiral are when the cells divide and they rotate. I don't know how I got that. Anyway, I got it right this time. So the radial cleavage patterns is the cells divide and they're straight up, so they're right up above each other. And spiral cleavage is when the cells divide, they rotate. Okay, so protostomes exhibit spiral cleavage. It's going to be, our protostomes are going to be the flatworms and then the roundworms. The mollusks are the like clam and octopus, squid, and the annelids are the earthworms. Okay, and then deuterostomes exhibit radial cleavage in their embryo, where the cells divide and go straight up. Echinoderms are the starfish. They're due to this, they're closely related to us. Our group chordates are all of us, all the fish and mammals and birds and reptiles and amphibians. Okay. So we're still going on with protostome versus deuterostome. And here's another one of those protostome deuterostome features. Protostomes, their embryonic tissues are said to be determinate. They're determined early. So the cells differentiate and become certain specialized cells early on in the embryonic development. Determinate. They're already determined what they're going to be. Whereas in a deuterostome, an indeterminate development, the cells don't specialize yet. They go through some dividing, whether to, and then it would be that radial cleavage dividing. They go through that, but they don't change yet, and they don't differentiate into any specific tissues yet. So that's why we can have identical twins. That ball of cells can get bigger and then split, and the cells haven't differentiated yet. And so this ball of cells can become a whole human being, and this ball of cells can be a whole human being, and they both have the same DNA as one another. And so this one grows up into a perfect human being. This one grows up into a perfect human being. You know what I mean? And they are identical because they came from the same cell original cell. Indeterminate cells. I think I'm going to adjust this just a little bit. It's just off the, there we go. It's off the side here. So deuterostomes have indeterminate development. And that's another protostome, deuterostome feature within is let's see, there's this, the body cavity structure. What is the type of body cavity, or if they have one? So a coelom is a body cavity. An organism which is an acoelomate, what does the A mean? It means without. So that means without body cavity. So there are some organisms that don't have a body cavity. Now that's the digestive system there. You can see, let's use your pointer. There's the digestive system, but there's not a body cavity out here. There's just tissue here. So that's a solid flatworm. The body's solid. There's not a hollow cavity inside, besides the gut. So that is an acoelomate. And then it's pseudocoelomates, pseudo means false, false body cavity. They have a body cavity, so they're a little more advanced. That body structure is a little more advanced than the acoelomates. But now what they have, so they have this body cavity here, and then there's a lining on the body wall. There's a lining out here that surrounds that body cavity. Now, a eucoelomate, a true coelomate, true body cavity organism, which is more advanced, has this. You see here, they've got a lining on the outer body wall, and they have a lining covering the inner gut area, covering that too. So you've got a red lining here and a red lining here. You've got a stronger supported coelom because you've got an inner lining and an outer lining. But we don't have that inner lining on these. You see the difference? We don't have that inner lining on the pseudocoelomates there. We just have the red outer lining. We don't have that inner lining. Okay. So there's advancement from an acoelomate to a pseudocoelomate and a eucoelomate. Right. Other animal features that they can use to determine relatedness. Here's one, metamerism, easier known, better known, or I think easier known as segmentation. So the question is the animal segmented or not? So here, you can see that we have a segmented earthworm. There's another, it's not an earthworm, but it's within the same group. It's an annelid. You see all the body segments there. And then here's all the segments that you see on a lobster, body segments. It's a repetition of body segments. Each segment is a metamere. Each segment is termed a metamere or a somite. Always more than one term for the same thing. Arranged along the longitudinal axis. The longitudinal axis, they're arranged along that. Annelids, that's the earthworms. Arthropods, that's the lobsters and insects. Chordata, that's the fish, amphibian, reptile, bird, mammal, primate. It's the chordates. Mollusks and echinoderms. Mollusks, the clams, and echinoderms, the starfish, they're not segmented. Okay, so is the body plan segmented or not? Another question determining relatedness. And then here's I believe this is our final one. Yeah, this is our final one. Does another feature, this is outside of the protostome deuterostome thing. This feature was outside the protostome deuterostome thing also. It's just another feature in general you could look at. And same with this. This is just another feature in general you can look at. Do they have a head or not? Cephalization. Development of a distinct head. A head is typically a concentration of nervous tissue and sensory organs in a common place, mainly found in the bilaterally symmetrical animals. The advantage is that it's facing the direction the animal is moving into the environment. You've got all your sensory organs directed in the direction that you're going, right? You've got your eyes looking this way. You can smell what you're getting into. I suppose you can taste it. You can hear what you're going into. You've got all these sensory organs that can detect things as you approach your environment. They're aggregated into a head region that you direct forward. Cephalization, isn't that beautiful? I mean, you look at that, look directed forward. The ears, I never realized it, but the tiger's ears, I never got close enough to one to see that. But the ears are directed forward, the eyes are directed forward, the nose is directed forward, the mouth and the teeth are directed forward. What's that animal? Time out. Hey. I mean, I'm just telling you what okay, animal. Honey. What animal? Here. Okay, honey. Oh, go see mom. Go see mom, hon. Okay, go see mom, honey. Well, I think I'm almost done here. Um, Jaden. Okay. Well, got that. Pardon me. So I mentioned in the first video I have twins. Well, there's the other one. The other one came in. Okay. So here we go. Let's see what do we got. So we're still on our head. And like I said, okay, so you've got your teeth and taste and directed forward. You got your nose directed forward, your eyes forward, your ears. Wow, beautiful. And that's their last one for this introduction. Here's some of the features that they use that they know arose um, earlier or later in evolutionary time, and they've used those to kind of separate the organisms. Okay, these we went this way, we developed this, and these went this way, and we developed that, and these went that way, branching things. So let's look at we've got multicellularity. Let's see here. Let's make this. Let's get that. Make sure it's clear. Okay, so let's see, multicellularity. No germ layers in an embryo. No true tissues developing because of no germ layers develop. Then you have a little bit more advanced thing. You have germinative layers developing, and then tissues from that. So there's a separation. Then over here, if we follow this, let's use our pointer. We got another separation. Let's see, right here, we've got a branch right here. Two germinative layers and radial symmetry. Two germinative layers and radial symmetry. So that's a cnidarian, huh? Yeah, a cnidarian, the jellyfish. And then you go this way. Now we've got three germ layers, and the organisms typically develop into bilateral symmetry. Okay, so that's all the rest. So this is going to be the cnidaria. So we don't look at the ctenophores or ctenophora. We don't look at those. We look at cnidaria. And then let's see what we have next. We have the development of a body cavity. These folks don't have a body cavity. Acoelomates, that'll be the platyhelminthes. So that's the flatworm, and the roundworms actually are. We don't look at the ribbon worms. Okay, so we look at the flatworms. So that's the organism we're going to see that has that is an acoelomate. And then you go this way. Now we're going to have all these other ones here. We'll look at the roundworm and mollusk and annelid, arthropod, echinoderm, chordate. Those all are then coelomates. You know, the roundworms being pseudocoelomates, and all the others that we're going to look at will be eucoelomates. You see, so this is a kind of a good slide to show the advancement through time, the features that developed through time. Then once we have these coelomates, now we have the potential of being a protostome or a deuterostome. Protostome was the organisms in which the first blastopore becomes a mouth. It's going to be your mollusks and annelids and arthropods or protostomes, these guys. And then the deuterostomes, the deuterostomes we'll look at will be first the echinoderms, the starfish, and then all of us. The chordates is a whole unit of its own. The chordates. Um, the chordates are, like I said earlier, they, first you start with a fish, and then an amphibian, then a reptile, then you look at a bird, and then a mammal, and then within the mammals, a primate. That's our course. Okay, so I think that's the final slide here. There's your favorite slide, right? Okay, that's the introduction to zoology. Thanks for coming.